Hello, this overview covers plant cells. Before watching this video, please watch the animal cell overview video. Most of the structures that we went over in that video are also included in plant cells. In this plant cell, you can see typical eukaryotic cell structures, such as a nucleus, endoplasmic reticulum, ribosomes, Golgi, mitochondria, a plasma membrane, cytoplasm, and various vesicles. The largest structure in a plant cell is known as the central vacuole. We learned about vacuoles in the animal cell video. They are larger than vesicles, and they function in water balance and storage of water, salts, pigments, and toxins. Central vacuoles function to keep flexible plants upright, such as roses. They use a process called osmosis to create turgor pressure. The cell collects fluid in the central vacuole, which pushes the cytoplasm to the sides and up against a rigid cell wall. You'll learn more about this in the osmosis and diffusion lab. Plants are also noted for their cell walls that lie just outside the plasma membrane. Bacteria, fungi, and some protists also have cell walls. The cell wall protects and supports the cell. Cell wall composition varies depending on the type of cell. For plant cells, it is mostly composed of cellulose and linin fibers. The cell wall is the reason for the uniform shape of plant cells. When you look at the Elodea plant in lab, you will see the cells laid out in nice, neat rows. Plant cell walls have plasmodesmata, which are cytoplasm channels for communication between cells. When you look under the microscope at a plant cell, you will see little green structures. They are the most notable feature in a plant cell because they are green, not colorless like most cell structures. These are chloroplasts. They function in photosynthesis. They are similar in structure to mitochondria. They have an outer and an inner membrane, and they have a fluid-filled space called the stroma. Within the stroma, there are stacks of a special membrane called the thylakoid. The thylakoid membrane is folded and stacked up so it looks like rolls of quarters. Each stack of thylakoid membrane is called grana, and they are interconnected. Thylakoid membranes contain a green protein pigment called chlorophyll. Chlorophyll traps sunlight energy used in photosynthesis. Just like mitochondria, chloroplasts contain their own circular molecules of DNA and ribosomes. We know this means they can make some proteins. Besides chloroplasts, there are other plastids, such as chromoplasts, which contain pigments like lycopene that makes tomatoes red, and leucoplasts that store starch, oils, and proteins. For example, amylioplasts in potatoes store starch. Now that you know about mitochondria and chloroplasts, we can learn about the endosymbiotic theory. This theory explains how mitochondria and chloroplasts were acquired in the evolution of eukaryotic cells. Evidence supports the idea that mitochondria and chloroplasts were individual prokaryotic organisms that were engulfed by much larger cells. This created their double membrane structure. Some early cells engulfed prokaryotic cells that could make ATP. Instead of killing the engulfed cell, it was kept alive to provide the host with ATP, and the host cell provided the engulfed cell with protection. We call this a symbiotic relationship. Both organisms benefit. Endo means inside, so the word endosymbiotic means a mutually beneficial relationship where one being is inside of another. The same thing happened to prokaryotic cells we call cyanobacteria because they can make their own carbohydrates. These cyanobacteria became chloroplasts, and the ATP-producing prokaryotic cells became mitochondria. The evidence which supports this theory includes the facts that chloroplasts and mitochondria are both similar in size to bacteria. They both have a double membrane. They both have their own DNA and ribosomes, and they're both able to replicate themselves. Thank you for watching this overview.